So now we're back in the paleontology lab at the Las Vegas Natural History Museum. So this is where we take the sediment once it's screen washed. From there, we use our microscopes to pick through the sediment and pull out any bones, teeth, or shells that we find there. So these vials hold very small fossils, which are what we pick out of the sediment once we screen wash it. So that can be things like teeth or shells. So as graduate students, a lot of the time we take on additional smaller projects in addition to our larger dissertations. So I'm taking on a smaller project looking at the microvertebrates at Tule Springs. So that's things like rodents, fish, reptiles, amphibians. And I'm looking at those because very small animals will live in very specific types of environments. They're really sensitive to their habitats, whereas larger animals can move around, eat various things, and they won't be so sensitive. So these small animals will give us very good indicators of what type of environment there was at Tule Springs.
So the side project that I'm working on is actually describing the ostracods in the sediments of Tule Springs. And so the ostracods have very, very small shells, and they're actually crustaceans, so they're related to crabs and shrimp. And their shells are actually preserved in the fossil record very often. And if the species found at Tule Springs are still alive today, then we know exactly what types of water conditions that they were living in. So we can really pinpoint the surface temperature, the pH, even the salinity of the water. So it'll help us really kind of pinpoint exactly what sort of environment that was available to aquatic animals at Tule Springs.